Welcome to Module 2 of Supporting Students with Dyslexia, the Brain Science of Dyslexia. In this module, we will learn about the neurological basis of dyslexia and the regions of the brain involved in dyslexia. We have a lot of learning ahead of us, so let's get started. Professional goals and learning objectives. We will engage in the Module 2 of Supporting Students with Dyslexia with the following professional goals and objectives in mind. Enhance educators' understanding of the neurological basis of dyslexia. Gain a better understanding of the brain structure as it relates to dyslexia. Discuss the phonological processing deficits and genetic and hereditary factors related to dyslexia. Recognize the compensatory mechanisms employed by dyslexic individuals. Promote evidence-based practices for teaching dyslexic students based on brain science insights. Equip educators with knowledge about the brain regions involved in reading and language processing. Understanding the neurological basis of dyslexia. The neurological basis of dyslexia is a complex and well-researched area that involves understanding how the brain processes language and reading. Here is an overview of the neurological basis of dyslexia. Brain structure differences. Phonological processing deficits. Genetic and hereditary factors. Compensatory mechanisms. And plasticity and intervention. Let's look at each neurological aspect of dyslexia in detail. Brain structure differences. Studies using neuroimaging techniques such as functional magnetic resonance imaging, fMRI, and structural MRI have revealed that individuals with dyslexia often have structural and functional differences in their brains, particularly in areas associated with reading and language processing. Research into dyslexia has indeed identified differences in brain structure, particularly in the areas associated with reading and language processing. Here are some key findings related to brain structure differences in individuals with dyslexia. Planum temporale asymmetry. The planum temporale, a region in the posterior part of the left temporal lobe, is known to play a crucial role in language processing, including phonological processing. Research has shown that in typical readers, there is a leftward asymmetry in the planum temporale, with the left hemisphere being larger. In some individuals with dyslexia, this leftward asymmetry may not be as pronounced or may even be reversed. Gray matter abnormalities. Studies using structural MRI have identified differences in gray matter volume in several brain regions in individuals with dyslexia. These differences are often observed in areas associated with reading, such as the left occipitotemporal cortex and the left superior temporal gyrus. Cerebellar involvement. The cerebellum, traditionally associated with motor control, has been found to play a role in language and reading. Some research has shown that individuals with dyslexia may have cerebellar abnormalities or atypical activation patterns in the cerebellum during reading tasks. Posterior parietal cortex. The posterior parietal cortex, which is involved in attention and visual processing, also shows differences in individuals with dyslexia. These differences may impact visual attention and the ability to rapidly process visual information during reading. Broca's area and angular gyrus. Differences in the left inferior frontal gyrus, commonly known as Broca's area, and the left angular gyrus have been observed in individuals with dyslexia. These areas are crucial for language and phonological processing. White matter pathways. Dyslexia is associated with white matter abnormalities, affecting the integrity of neural pathways that connect different brain regions involved in reading and language. These abnormalities may impact the efficient transmission of information within the brain. It's important to note that while these brain structure differences are associated with dyslexia, they do not serve as diagnostic markers for the condition. Dyslexia is typically diagnosed based on a combination of factors, including reading assessments, behavioral observations, and standardized tests. Furthermore, the brain is highly adaptable, and early intervention and targeted reading instruction can lead to changes in brain structure and function that support improved reading skills in individuals with dyslexia. Phonological processing deficits. One of the key neurological features of dyslexia is a deficit in phonological processing. This refers to the ability to recognize and manipulate the individual sounds of spoken language, phonemes. Dyslexic individuals may have difficulty distinguishing and processing these sounds, which are essential for reading and spelling. Phonological processing deficits are a central and well-documented aspect of the neurological basis of dyslexia. Research has extensively explored the role of phonological processing deficits in dyslexia, and here are some key findings within this context. Phonological awareness deficits. Phonological awareness is the ability to recognize and manipulate the individual sounds, phonemes, in spoken language. Research consistently shows that individuals with dyslexia often have difficulties with phonological awareness tasks. This includes tasks like rhyming, segmenting words into their individual sounds, and blending sounds to form words. Impaired phonological memory. Phonological memory refers to the ability to temporarily store and manipulate phonological information. Individuals with dyslexia may exhibit deficits in phonological memory, which can affect their ability to remember and manipulate speech sounds, making it challenging to learn new words and spell them correctly. Difficulty in decoding. Dyslexia is characterized by difficulties in decoding words, which involves applying phonological knowledge to recognize and pronounce written words. The phonological processing deficits in dyslexia can impede the development of this critical reading skill. Underactivation. Functional neuroimaging studies using techniques such as fMRI have revealed differences in brain activation patterns in individuals with dyslexia during phonological processing tasks. These differences often involve underactivation in key areas of the brain responsible for phonological processing, such as the left inferior frontal gyrus and the left superior temporal gyrus. Atypical brain connectivity. Dyslexia is associated with atypical functional connectivity in the brain, particularly in regions related to phonological processing. This atypical connectivity can affect the coordination of neural activity during phonological tasks. Genetic and hereditary factors. Research has identified specific genes associated with dyslexia that are related to phonological processing and brain development. These genetic factors may influence the development of neural networks involved in phonological awareness and reading. Genetic and hereditary factors. Research has identified a genetic component to dyslexia. Specific genes related to brain development and language processing have been linked to an increased risk of dyslexia. These genetic factors can affect the development of neural networks involved in reading. Genetic and hereditary factors play a significant role in the neurological basis of dyslexia. Research has consistently demonstrated a strong genetic component to dyslexia, highlighting the influence of specific genes on brain development and language processing. Here are key findings related to genetic and hereditary factors in the context of dyslexia. 1. Familial clustering. Dyslexia tends to run in families, and individuals with a family history of dyslexia are at an increased risk of developing the condition. This familial clustering suggests a hereditary component. 2. Heritability estimates. Twin and family studies have been instrumental in estimating the heritability of dyslexia. These studies indicate that genetic factors contribute significantly to the risk of developing dyslexia. Heritability estimates typically range from 40% to 70%. 3. Identification of candidate genes. Advances in molecular genetics have led to the identification of several candidate genes associated with dyslexia. These genes are involved in various aspects of brain development, particularly in regions relevant to language and reading. 4. FOXP2 gene. The FOXP2 gene, implicated in language development, has been studied in the context of dyslexia. Variations in the FOXP2 gene have been associated with language impairments, including difficulties in phonological processing, a core challenge in dyslexia. 5. DCDC2 and DYX1C1 genes. Variants of the DCDC2 and DYX1C1 genes have been linked to dyslexia. These genes are involved in neuronal migration and the development of brain structures critical for reading, such as the left temporoparietal cortex. 6. ROBO1 gene. The ROBO1 gene has been associated with dyslexia and is involved in neural connectivity. Variations in this gene may impact the formation and function of neural pathways relevant to reading. 7. Hereditary transmission patterns. Dyslexia is inherited in a complex manner. Multiple genes are likely involved, and the interplay of genetic factors contributes to the variability in the expression of dyslexia within families. 8. Gene-environment interactions. While genetic factors play a substantial role, interactions between genes and environmental factors also contribute to the development of dyslexia. Environmental influences, such as early language exposure and educational experiences, can modify the expression of genetic predispositions. 9. Polygenic nature. Dyslexia is considered a polygenic trait, meaning that multiple genes contribute to its manifestation. The complexity of dyslexia's genetic basis involves the interplay of several genetic factors, each making a modest contribution to the overall risk. 10. Genetic overlap with other conditions. There is evidence of genetic overlap between dyslexia and other neurodevelopmental conditions, such as attention deficit/hyperactivity disorder, ADHD, and specific language impairment, SLI. Shared genetic factors may contribute to the co-occurrence of these conditions. Understanding the genetic and hereditary factors in dyslexia is essential for advancing our knowledge of its neurobiological basis. This knowledge can inform the development of targeted interventions and support strategies tailored to individuals with dyslexia based on their genetic profiles. Compensatory mechanisms. Individuals with dyslexia often develop compensatory mechanisms to navigate the challenges associated with reading difficulties. Research on the neurological basis of dyslexia has identified various compensatory strategies at both the behavioral and neural levels. Here's an overview of what research says about compensatory mechanisms in the context of dyslexia. 1. Recruitment of additional brain regions. Neuroimaging studies have shown that individuals with dyslexia may recruit additional brain regions beyond the typical reading network to compensate for difficulties in phonological processing. This recruitment can involve areas associated with visual processing, executive functions, or other cognitive processes. 2. Increased activation in frontal regions. Some individuals with dyslexia exhibit increased activation in frontal brain regions, such as the prefrontal cortex. These areas are associated with executive functions, working memory, and attention. The heightened activation may reflect an increased reliance on these cognitive processes to compensate for phonological deficits. 3. Activation of right hemisphere regions. While reading is typically left lateralized in the brain, individuals with dyslexia may show increased activation in the right hemisphere regions. This includes areas involved in visuospatial processing. The right hemisphere may play a compensatory role in supporting reading-related tasks. 4. Enhanced visual processing. Compensatory mechanisms often involve enhanced reliance on visual processing strategies. Some individuals with dyslexia may use visual cues, such as relying more on the appearance of words or memorizing the shapes of words, to compensate for difficulties in phonological decoding. 5. Multimodal processing. Compensatory strategies may involve engaging multiple sensory modalities. For example, individuals with dyslexia may benefit from simultaneous visual and auditory input when processing written information. This multimodal approach can provide additional support for comprehension. 6. Whole word reading strategies. Some individuals with dyslexia adopt a whole word reading strategy, where they recognize and memorize entire words rather than decoding them phonetically. This strategy can be effective for reading familiar words, but may pose challenges with unfamiliar or non-phonetic words. 7. Strategic reading approaches. Compensatory mechanisms often include the development of strategic reading approaches. This may involve using context, predicting content, and relying on contextual cues to infer the meaning of words. Strategic reading helps compensate for difficulties in decoding. 8. Behavioral adaptations. At the behavioral level, individuals with dyslexia may develop compensatory strategies such as slowing down their reading pace, rereading passages, or using aids like finger tracking to maintain their place in the text. These adaptations can enhance comprehension and accuracy. 9. Self-regulation strategies. Some individuals with dyslexia develop self-regulation strategies to manage frustration and maintain motivation. These strategies may include setting realistic goals, breaking tasks into manageable steps, and seeking support when needed. Plasticity and intervention. The concept of neuroplasticity, the brain's ability to reorganize itself and form new neural connections, is particularly relevant within the context of interventions for dyslexia. Research has shown that targeted interventions can lead to structural and functional changes in the brain, indicating that the neurological basis of dyslexia is amenable to positive modifications. Here's what research says about plasticity and intervention in the context of dyslexia. 1. Neural changes with intervention. Studies using neuroimaging techniques, such as functional magnetic resonance imaging, fMRI, and structural MRI, have demonstrated changes in brain structure and function following dyslexia interventions. These changes are often observed in areas associated with reading and language processing. 2. Increased activation in reading regions. Effective interventions for dyslexia are associated with increased activation in brain regions implicated in reading, such as the left temporoparietal cortex. This heightened activation is indicative of improved neural efficiency and more effective processing of written language. 3. Enhanced connectivity. Interventions can positively impact functional connectivity between different brain regions involved in reading. Improved coordination between these regions enhances the integration of phonological, visual, and linguistic information during reading tasks. 4. Impact on white matter integrity. Research suggests that interventions can lead to improvements in white matter integrity, addressing abnormalities observed in the white matter tracts of individuals with dyslexia. Enhanced white matter connectivity contributes to more efficient communication between brain regions. 5. Longitudinal studies. Longitudinal studies tracking individuals with dyslexia before and after intervention have provided insights into the dynamic nature of neuroplastic changes. Positive alterations in brain structure and function may continue to evolve over time with sustained intervention. 6. Age-related plasticity. While there is evidence of neuroplasticity in response to interventions across various age groups, there is also a suggestion that the degree of plasticity may vary with age. Early intervention is often emphasized to leverage the heightened plasticity observed in the developing brain. 7. Differential effects of interventions. Different types of interventions may elicit distinct patterns of neuroplastic changes. For example, interventions targeting phonological processing may lead to alterations in areas associated with phonological awareness, while interventions focusing on visual processing may impact visual regions. 8. Transfer effects. Some interventions show transfer effects, indicating that improvements in specific reading skills generalize to broader aspects of reading and language processing. This suggests that targeted interventions can have a holistic impact on the reading abilities of individuals with dyslexia. 9. Individual differences in plasticity. Individual variability in response to interventions is recognized. Factors such as the severity of dyslexia, the presence of comorbid conditions, and the specific profile of cognitive strengths and weaknesses can influence the degree and nature of neuroplastic changes. 10. Maintenance of gains. Sustained gains in reading skills following intervention are associated with the maintenance of neuroplastic changes. Continued practice and reinforcement of skills contribute to the consolidation of positive alterations in neural networks. Understanding the neuroplasticity associated with dyslexia interventions is crucial for refining and optimizing intervention strategies. Research in this area informs the development of targeted, evidence-based interventions that capitalize on the brain's capacity for change and adaptation. Brain regions involved in reading and language processing. Click on the Resources button on the top to download and view a detailed list of brain regions involved in reading and language processing in dyslexia. The information in the table identifies the region of the brain, its function, and its dyslexic involvement. Key takeaways. In conclusion, the brain science of dyslexia provides educators with valuable insights into the neurobiological basis of dyslexia. We've explored the intricate workings of the brain regions involved in reading and language processing, gaining a deeper understanding of how dyslexia impacts these processes. By delving into the structural and functional aspects of the dyslexic brain, we've learned about the complexity of this condition. Recognizing the genetic and hereditary factors that contribute to dyslexia's neurobiology has highlighted the importance of early identification and intervention. We've also discussed how dyslexic individuals employ compensatory mechanisms to navigate their challenges. Importantly, we've connected this knowledge to practical applications in the classroom, emphasizing evidence-based strategies for teaching dyslexic students and fostering effective communication and collaboration among educators, parents, and support services. Next section, Early Signs and Identification. In the upcoming section, we will shift our focus to identifying the early signs of dyslexia and understanding the importance of early intervention. We will explore strategies for recognizing dyslexia in students during their academic journey, ensuring that timely support is provided to enhance their reading and language skills.